Hi, this is John Christianson. Thanks for taking a couple of moments to learn about undergraduate research opportunities in the Christianson Lab at Boston College. If you don't listen to the rest of this slide, you can learn a lot more just by going to christiansonlab.bc.edu or emailing me at john.christianson.2@bc.edu. All right, research experiences in our lab tend to be focusing on three different questions. The first one is, how does stress influence behavior? We do this in several different ways. Sometimes we're looking at the effect of stress directly experienced by rodents, rats usually, on their brain function, on their behavior, because we're trying to figure out what are the mechanisms of stress vulnerability and stress resilience as they relate to conditions like post-traumatic stress disorder and depression. More recently, we've been looking at how social sources of stress, like interacting with another rat who's just experienced stress, affects the behavior of those who are observing that stress. Appropriate responses to social stress signals are really important for developing empathic relationships and demonstrating helping behavior and so on, and simply just reading the emotions of other people. In light of that, we're interested in how the brain makes social decisions, like who to help, who to avoid. These things are kind of important questions at the moment while we're all practicing social distancing. So we use a variety of methods. Many of them right now are focused on understanding the structures of the insular cortex and its connections to other areas in what we call a social decision-making network of brain structures. So we can use drugs to affect that brain region, use the viral delivery of tools like optogenetics and chemogenetics, which let us control specific types of cells within the brain at very specific time points, and cells with specific tracts or projections to other brain regions within these networks, while rats are experiencing stressful or social choices, and we can learn a lot about how those brain regions relate to behavior. Our last question is sort of a broad one, but in the social context, we're interested: how do neurons and in the circuits that they're in encode emotion? So we can do that in a few ways. This is one of the cool ones that you can see in this video. The rat with a cable coming out of its head has a fluorescent gene in its insular cortex that changes how bright it is when those neurons are active. It's called GCAMP7F. In the trace that you're seeing above is the relative fluorescent change as the experimental rat spends time interacting with the target one. So anytime there's a rise there, neurons, in this case in the insular cortex, were more active. So ongoing research projects are utilizing this technique and several different behavioral types of tests to understand how neural circuits are encoding emotion. Overall technologies that we use in the lab include behavioral tests, surgical manipulations, biochemistry analyses of products of neural activity in the brain, a lot of microscopy to look at brain function and neuronal activities, the anatomy of various tracts, and then, like I said, chemogenetics, optogenetics, and pharmacology, the use of drugs to try to understand how specific chemicals or specific circuits are involved. So if this is interesting to you, there's lots of ways to participate. Undergraduates can take the course Psych 2205 called Undergraduate Research, which is a one-semester course which would fulfill several requirements in the different psychology and neuroscience degrees. There are paid positions, which include undergraduate research fellowships, which I can apply for if you're interested, which would pay you a small stipend for your work. If you're eligible for work study, you can perform some of your work study hours in the laboratory. More advanced students might conduct a senior thesis or an honors thesis. Typically, those students would have worked in the lab for at least one summer, maybe a whole semester or a whole year prior to develop a project that's interesting to them. And excuse that beep. And then there's summer research fellowships, which are also paid positions. So what should you do? Well, most of the students who have worked in the lab will have taken Psych 2283, Behavioral Neuroscience. You need to have some background in neuroscience and biology and behavior to get the most out of this experience. If you haven't taken it, that doesn't mean that you're not eligible to work in the lab, but it is something that will give you, I guess, sort of better preparation. You should also take a look at your schedule. Research in neuroscience tends to require chunks of time. So usually we're looking for students to have a few hours free a few times a week in their schedule so you can make meaningful contributions. The next thing you should do is contact me, j.christianson@bc.edu, and describe to me in an email what your goals are for research experience, what courses you've taken, and any questions that you have, what kinds of topics based on what I just showed you might be interested in. You should look at christiansonlab.bc.edu to take a look at our recent publications and see kind of who's on our team. You might have had some of our students as TAs or fellows in your classes, so you can ask them about what's going on. I will end by saying research experiences in our lab are limited in a number of ways. We have a limited number of space, and it also requires a good deal of kind of safety training before you can do this research. So we're looking for very serious students who want this experience, and it's best to get in touch with me early in your career so that I can make sure that you're in a position to get into the lab, because if you kind of reach out maybe in your junior or senior year, it could be that there's already a waiting list. So with anything, I just get in touch, and I look forward to talking to anybody who's interested in lab experience, and hopefully we can get you involved. Take care.